I'd like for you to take a moment and imagine a cell. It can be any cell you choose. It can be a neuron or brain cell or even a skin cell. Now I want you to imagine that cell like a mini manufacturing plant. Now that sounds silly, doesn't it? But if you think about it, they do a lot of similar things. One of those things being production. Now, no, cells don't produce goods such as Hasbro, who produces toys, or Apple, who produces computers and other technologies, but cells produce things that they need to survive. So cells produce things called proteins, and a lot of those proteins are utilized for many different cellular processes. But also, like a manufacturing plant, cells have to undergo shipping and receiving because sometimes those products that are made in a cell or proteins are necessary for other cells that are nearby. And so they have to ship those proteins out. And sometimes they have to receive proteins too to aid with their own cellular needs. But something that a lot of people don't think about is, what does a cell do with the waste? A lot like a manufacturing plant, that's come into light recently is, what do manufacturing plants do with all of their waste? Now some have started the recycling, and it easily translates to, well, how does a cell accommodate for the waste that is a byproduct of their production? Well, cells have a number of different methods of dealing with this protein waste. And one of those methods is through the ubiquitin proteasome system. Now, you might be thinking, wait, if a cell produces these proteins for their survival, then why are they waste? And that's a really good question. But sometimes proteins are produced for a single purpose, and they may not get damaged or degraded meaning destroyed, in that process. So sometimes you still have leftover proteins that need to be taken care of. Other times, as I mentioned, proteins get damaged. Cellular environment's a little rough and can damage proteins pretty easily. And other times, there's a problem with production, and you're not producing the thing that you're supposed to. And so proteins then are non-functioning, they don't fold properly, and those ones need to be taken care of just as importantly as the other ones. And the problem in a cell is, if these proteins don't get taken care of as waste, or cleared as waste, then it can cause a lot of problems. Those bad proteins can form things called aggregates, which means that these proteins come together and form these complexes that can actually be really toxic to a cell. At that point, sometimes the cell has to undergo something called apoptosis, or programmed cell death, which isn't always a good thing, especially if you imagined a neuron. Neurons undergoing apoptosis isn't always the best thing. It's better than nothing, but not always the best choice. So, as I mentioned earlier, the ubiquitin proteasome system aids to clear those proteins before they become aggregates and become toxic to the cell. So, what is a proteasome? Well, a proteasome is a barrel-like structure. It's made up of rings, and in the very center of the barrel, and in the middle of those rings, you can imagine are almost like these scissors. And what the scissors are responsible for doing is something called proteolytic cleavage. Now, what does that mean? Well, proteolytic cleavage just means chopping up bad proteins, or unnecessary proteins, or damaged proteins, whichever is being targeted to be degraded or eliminated as waste. So you can almost think of the proteasome as a mini recycling bin, or a mini garbage disposal. It's responsible for taking a protein, unfolding it, chopping it up into little pieces, and taking those little pieces and recycling them so more proteins can be made later. Now, because of its proteolytic responsibility, the proteasome is tightly regulated. At the end of either end of the barrel are these little gate-like structures that require things called activators to open. Now, the one that I mentioned earlier, the ubiquitin proteasome system, has a regulator on either end that requires energy, as ATP, and it can recognize proteins that are targeted for the proteasome, unfold them, and actually pull them through that barrel after the gate's been opened and causes them to get all chopped up. So that's a little bit about the proteasome. But what happens when the proteasome can't degrade these proteins or chop them up? What happens then? Well, those aggregates that I talked about can happen. Those things that are super toxic for a cell can happen. And what happens when certain proteins are unavailable to a cell, meaning the cell doesn't make them anymore? And what if those proteins are really important for activation of the proteasome? Well, that's what I study. I study dysregulation of a certain activator of the proteasome that doesn't require ATP and is responsible for being overexpressed, or overexpression meaning it's made too much in a cell. And when it's overexpressed, or is too much of it is being made, it can lead to certain types of cancers, such as colorectal, esophageal, cervical, and even ovarian. But what happens when that same activator still doesn't require any energy, isn't really being made at all, or it's something called underexpression? Well, when it's being underexpressed, that can help aggregates that are responsible for certain types of neurodegenerative disorders, or diseases that cause problems with your nervous system. And some of you might be familiar with those, such as Parkinson's, or Huntington's disease, or even certain dementias, such as Alzheimer's. What I study specifically is a poly-Q disorder, which means that a protein has a lot of certain types of amino acid that is really hard for the protein to degrade by itself. And it's called Huntington's disease. And as most of you know, it's a really devastating disease, mainly affects women, it can affect men, and it's onset. You don't really understand that you have it until you start showing symptoms at roughly 40 to 50 years of age. And at that point, if a person has that disease, they're more likely to pass it on, they are going to pass it on, to their offspring. And so I study that in regards to the underexpression of that regulator I was talking about, the energy-free one, and how, if we could restore normal expression, we could help clear those really bad aggregates that I was talking about previously in that specific disorder. And then hopefully we could find a potential drug treatment to help people with Huntington's disease and restore normal proteasome activity to clear all those toxic aggregates so they no longer suffer the effects of neurodegenerative diseases such as Huntington's.